Our lab historically has focused on developing tests for cancers of the blood. So that would be like acute myeloid leukemia, myelodysplastic syndromes, lymphoblastic leukemias.
And the test is a success.
It's a single test, gives you all the information you need. We can turn it around in as little as 3 days. It's really the first comprehensive single assay for risk stratification and diagnosis in myeloid neoplasms.
Dr. Eric Dunkevich is a professor of pathology and immunology, and he's the director of the Division of Genomic and Molecular Pathology at Washington University School of Medicine in St. Louis. The testing for blood cancers is performed on the medical campus in the McDonald Genome Institute, where the scientists were the first in the world to demonstrate whole genome sequencing for identifying genetic errors responsible for the development and progression of cancer. Now, this whole genome sequencing test for blood cancers is designed to identify the full suite of genetic changes in a patient's cancer cells. The test is called ChromoSeq.
Well, Eric came up with the name, and it's a rather clever name. ChromoSeq means a fusion of two words, chromosome, so this is a whole chromosome test, and sequencing. So this is the sequencing of the entire DNA in the chromosome. So it's a clever combination there. It is a blood test, but what it actually tests out of the blood are cells that are in the blood, and what can be quite rare malignant cells in the blood.
Our thought was, several years ago was, we're doing all this different testing for acute myeloid leukemia, different testing modalities. This makes it very complicated, but can we use whole genome sequencing as a single assay to replace a lot of that? About 270 different patient samples, we showed analytic performance. We showed that the accuracy that we could generate with whole genome sequencing, the information that we got was the same as what we would get from conventional testing. And then in addition to that, about 20% of patients had new findings that we could validate that were not detected by conventional testing, which now we can generate more sensitive data with whole genome sequencing, and that helps stratify how aggressive the therapy is.
And now, ChromoSeq is the first whole genome sequencing test for cancer to be approved by the Centers for Medicare and Medicaid Services, or CMS.
This test actually changes the way that patients are treated, and that's what CMS recognized.
Washington University oncologists have been using the test successfully since 2021.
It is able to provide much, much more detailed information about what's going on in that cancer cell than other types of tests.
And so we're running it primarily now for initial diagnosis. Is it an AML or is it a myelodysplastic syndrome? And so there are certain gene mutations that can tell us this is an MDS or this is an AML, and those are treated differently, different outcomes.
And with Medicare approval for the test, physicians nationwide caring for Medicare patients with blood cancers will be able to order the test through Washington University Pathology Services, and Medicare will cover the cost. Meanwhile, more trials are underway.
We have a clinical trial now that's currently enrolling here at Washington University Barnes-Jewish Hospital for ChromoSeq. And so in that trial, we're seeking to understand how the ChromoSeq assay and the information we get from that changes patient management and changes the physician's approach to those patients compared to conventional testing.
Because it's a whole genome test, we believe that this test will be useful with some modifications for virtually any kind of cancer eventually.
They are also experiencing success with another test called MyeloSeq.
It is also for blood-based cancers, and in this case it's for myeloid leukemias, which is why it's called MyeloSeq.
MyeloSeq is targeted. It looks at about 50 genes.
MyeloSeq is currently used for patients at Siteman Cancer Center after treatment following the ChromoSeq diagnosis.
We're using that assay now for most of our AML and MDS patients post-therapy to monitor what's happening. So MyeloSeq is very, very sensitive. Because we're only looking at 50 genes, we can look very deep. You imagine like a patient, we do ChromoSeq on them, we identify specific gene mutations in those patients, and then the patients are treated. And then the next thing that we want to know is, well, did the treatment help? Did it clear the malignancy? Did it clear the clones? And so what MyeloSeq can do is identify those gene mutations with a very, very high sensitivity, so several orders of magnitude more sensitive than ChromoSeq.